Presentation up, Tim. Let's check it's showing.
Yeah, I checked it with Tulio a few minutes ago, so it should be all right.
So I'm going to have the honor of introducing Tim tonight. And I met Tim when he was already director of the JK Centre, having been educated in the UK and done a stint in South Africa at De Beers Diamond. So good pedigree, having got a lot of training in South Africa, where great metallurgists come from. And working with Tim, I was in the P9 project. I was a novice. I was learning as I went, as he clearly knew, encouraged me to get more site experience. And I was really impressed with Tim's deep technical knowledge of all the broad range of projects across the JK. Really knew what was inside every single one. And that was really impressive for a director to have their hand around it all. You couldn't get past Tim bluffing him on any project at all. And also really pushed the industry connection, which the JK was famous for and based on from Alban's days and then Don, is the connection of getting knowledge into practice. And that just taught me immensely, that connection with the JK then. And so you really got these brilliant outputs and built brilliant people in that time. And they're kind of a legacy of legendary people who've come out of the JK in your period as director, Tim. And I'd say that Tim's presentations are always enjoyable and edifying. And bear in mind that Tim had told me that you should always have a question on every presentation you hear. So he's expecting everyone will have at least one question available. Thank you, Tim. Over to you.
Thanks, Malcolm. Well, hello, everybody. Thanks for the kind introduction, Malcolm. And yes, you and I have known each other a long time, and it's all been a pleasure. And hello to all of my old friends out there I haven't seen for a while, and to all of my new friends. Thanks to Aubrey for asking me to give this little talk, and to Tulio for organizing it. And thank you all for attending. I'm sure you had better things to do, but I hope your time won't be entirely wasted. Well, we all run experiments, including plant trials and lab tests, and we all analyze data. And the purpose of my talk today is very simple: to make the case that we should, as far as possible, design our experiments and analyze the data according to statistical principles, as other numeric professions do. I believe that stats should be in our DNA, routine, uncontroversial, and not something we use as a last resort to rescue lousy data from a poor experimental design. Well, I wonder how many of you will tell us that you include statistics in your skill set. I suspect most of you were like me. You did Stats 101 in maths in first year university, got 51% in the exam, and said, well, thank goodness I'll never have to do that again. Well, I'm going to explain why I don't think that's a good attitude. Designing and analyzing experiments using robust statistical principles allows the effects of the variables we're interested in to be estimated in the minimum number of runs. That limits the cost of the experiment, whilst controlling the risk of making the wrong decision. That's key. Not doing it right may get the wrong answer, no answer at all, or if you're lucky, the right answer, but take much too long. So why don't we do the right thing? And some of you do, I know, but not all of us do. So usually I think it's either ignorance or prejudice or both. So I'm going to use a few mineral processing examples to persuade you, or try and persuade you if you're not already persuaded, that doing it right is usually better, faster, and cheaper than doing it wrong. And it's much easier than you think. So let's get started. I'll just share my screen. Hopefully, there we go. Hopefully you're seeing a mountain. And so, Tulio, is that okay?
Yes, go ahead.
Okay. Well, look, I'll make no apologies for the rather apocalyptic title of this talk. It reflects the fact that with some honorable exceptions, as I said, most scientists and engineers in the mineral processing field are not as familiar as they should be with statistical methods. which is a pity because they'll greatly enhance the effectiveness, I think, of their experimental work. Things are better than they used to be 20 years ago, but we're not there yet. And I think this session I see as a small step on the road to good practice. The title also reflects a comment by someone who was probably the greatest statistician that ever lived. Seems to have decided this not to work. Hold on. This man, R.A. Fisher, who was a brilliant mathematician. He was a professor at Cambridge, but he also spent 20 years at the Rothamsted Agricultural Research Station in England, which was the greatest one in the world at the time. So he not only understood the mathematics and developed lots of the techniques we use routinely today, but he was also a very practical man. He was designing experiments for agricultural scientists who have the same problems as we do, difficult data and small samples. So he said this: to call in the statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment died of. So that's a plea for planning. And he wrote the first book on statistical methods for research workers, which, when I was preparing this talk, I realized by extraordinary coincidence was published exactly 100 years ago. So we can't say we weren't warned and that we didn't know. There have been, of course, thousands of great textbooks since then, but this was the first. And you'll see graphs and tables in it that look just like you can get out of Excel today. So let's agree on a few definitions. What is an experiment? Well, I think it's a process which results in the collection of data to test a hypothesis. If you're a PhD student, well, your hypothesis is what you set out to solve, but a hypothesis can be anything. What is the impact of a new control system on a SAG mill on throughput, for example? A hypothesis is that it's better. A good experiment is one which gets the right answer with the minimum of effort. And you'll notice we've already introduced the idea that experiments shouldn't be any longer than they have to be. A more technical definition, an efficient experiment is one which obtains optimal estimates of the effects of the chosen factors or variables in the minimum number of runs. The optimal estimate is a mathematical definition, as it turns out. So I don't think this should be a controversial statement. Let's do good rather than bad experiments. So what I'm going to run through quickly is three parts to this talk. First of all, what do others do? What can we learn from other professions? Then a little bit of a horror story on what we tend to do. And then finally, what could we do if we wanted to? So let's start with what others do. Well, talking of Fisher and his agricultural experiments, this is a classic experiment looking at, in a very large area, looking at crop yields as a function of the variety of the crop and fertilizer and things you can't control, like the fertility of the soil and microclimate and so on. You'll notice it's a factorial design, and that word will come up quite a bit in the next few minutes. Here's an interesting experiment on the effects of global warming on shallow freshwater lakes. So each of those tanks is simulating a shallow freshwater lake. It's a factorial design, 3 temperatures, 2 nutrient levels, and 4 repeats of the whole experiment so that you can capture the value of experimental error, which we'll talk about a bit later. This is a pharmaceutical study. This is a study of different possible medicines for the treatment of high blood pressure. This is an experiment that SciTech did some years ago, looking at the effects of temperature, solids concentration, particle size, and the choice of flocculant on supernatant clarity in settling tests. And you can see here straight away the nice, satisfying, symmetrical nature of the layout of the experiment. And yes, you've guessed it, another factorial. It says blocked on time. What this means is that the experiment is arranged without having to do any extra tests, such that any time effect, and time is a proxy for things that change with time, any time effect can be captured and measured and removed from the results. So that's a very powerful capability. This is a chemical engineering study of optimizing the yield of a chemical process as a function of pH and reactor volume. You see that little square in the bottom left-hand corner here. So that's a tiny factorial, and the whole purpose is to take the results of each little factorial experiment and, through analyzing the data appropriately, move the whole experiment up the contours, which of course, as an experimenter, you can't see. We're privileged to see them up until you get to the maximum point. That's a chemical engineering example. This is looking at optimum conditions for the production of biogas and other factorial, development of vaccines and other pharmaceutical products. Well, we all became experts on vaccine development, didn't we, during COVID? These again are done according to factorial design principles. And once you've developed a vaccine or a new medicine, it has to be tested in clinical trials with human volunteers. And this is quite interesting because no pharmaceutical company is permitted to sell or market a new drug unless the local relevant authority in Australia, it's something called the TGA, Therapeutic Goods Administration, unless the appropriate authority is satisfied that these trials have been done according to proper statistical criteria. So here, doing the right thing, and yes, you've guessed it, they look very like factorial experiments. Doing the right thing is actually mandated by government regulation. Now, the reason for that is it's to do with human health, and with human health, only the best will do. And these are the best way to go about doing experiments. Now, we don't have to put up with the government telling us what to do, but it seems to me a bit of a giveaway that they follow appropriate procedures. And I guess what I'm trying to say is we should do the same. So here's another example, manufacturing, looking at effective welding length and depth on a fabrication of a component. This is just showing you nice ways to visualize the outcomes of properly designed experiments. And this is one of my favorites I found in the Psychology Journal, experimental design of a strengths-based training to overcome academic procrastination. Well, I'm speaking to you as the world's greatest academic procrastinator, so I found this paper very interesting. But again, the point is experiments were done according to proper statistical principles, so proper conclusions could be drawn. And this is an example you're all familiar with, the idea that you can sample a population, in this case of people, and ask them questions like what's their favorite chocolate, or which political party would they vote for at the next election. And again, these sorts of exercises which we see reported in the media are all conducted according to proper statistical principles and correct sampling theory. So I hope I've been able to persuade you that everybody else does this stuff routinely. It's in their DNA. It's not controversial. And I think we need to follow their good example. So what do we tend to do? And this is a bit of a horror story, and it's not all like this, of course, but I want to make the point, emphasize it by making the point. So here's a couple of case studies. Here's a plant trial in a gold concentrator to look at the use of a new gold-specific flotation reagent. Could it give us a better recovery? Now, this was done according to the dreaded before and after idea. So you're running with the old reagent, the old condition, and you make a switch at an arbitrary time, and you put the new reagent in, and you take an average before, average after, and you look at the difference and make a decision. Well, here were the results. 70% with the old, 78% with the new, an improvement of 8%. Any of you who know anything about flotation plants and concentrators will know that that is too good to be true. And we all know that if anything's too good to be true, it probably is. So I was asked to referee a debate between the reagent supplier and the mining company, and it was the easiest money I ever earned because all I did was plot the data in time, and that's what you get. So along the x-axis, we've got about 3 months of data. Up the y-axis, gold recovery. Doesn't take a genius to work out that the gold recovery was increasing in the couple of months before the change anyway, and you could argue that this new reagent has actually stopped the improvement. So not only is it probably not better, it could actually be worse. Now, of course, the problem here is it's a terrible experiment. It's an experiment where you cannot unequivocally attribute an observed change in performance to the change you're trying to make. It's a very bad experiment. And here's another one which will please all the communitors out there, all you lot. This is a paper I found looking at the introduction of a fuzzy logic control system in a sag mill. So up the y-axis is tons per hour, time along the x-axis. And the authors of the paper actually drew a straight line to it. I think you would be forgiven for thinking that maybe we've got the same problem. There are ways to tease out trends in that data, and that might produce a better result. But at first sight, we have the same problem. The before and after can be influenced by other factors than the one we're interested in. So here's some experiments done with a 3-product hydrocyclone, which many of you will be familiar with. And what was required here was to look at the effect of two of the dimensions of the inner vortex finder, the length, which is LIVF on the x-axis, and diameter, DIVF, on the y-axis, the effect of those on the P80 of the final product to one of the products. Now, where you have a complex system, we're often enjoined to do one variable at a time, the one variable at a time method. So you pick a constant value of one of the variables, let's say LIVF of 200 mils, and then you do a number of experiments at different values of the other variable, DIVF. And then you make that one constant, and you do a number of tests at on the other variable, LIVF. And then you delude yourself into thinking you know about the effects of those two variables on the P80. Well, the true process contours for this particular quite complex process look like this. And you might wonder, well, how do I know that? Well, because the student who did this work did a great experiment, did a proper what's called a CCRD experiment, which produced a model that predicted the response surface for this quite complex device. And but of course, the one variable at a time achieves very little. It misses the optimum, which is in the top right-hand corner, and it's a very simplistic description of what is quite clearly a complex process. This is color-coded as usual, data set from a plant trial of a copper flotation reagent, again trying to improve recovery. So looking from left to right, it's a series of columns of date versus the corresponding copper recovery. Yellow denotes trial on, that means the new reagent. Green denotes trial off, that's the old reagent. And as you can see, what they tried to do was to run for a week on and then a week off and switch. The white ones are changeover days, which they basically rejected. Now, first of all, just in passing, why do it for a week? If you did it every day, you'd get the result 7 times faster. But that's another story. So there's about 5 months of data there. And at the end of it all they could really do was to take the average of the greens and the average of the yellow and see if they were different. And here was the result, about 0.5% difference in recovery. Now, you might say, well, that's really not worth anything. But as you all know very well, a large mine, 0.5% would be several $1 million a year, and therefore worth going for, if you could be satisfied that that change was statistically significant. And in fact, the people concerned used a little test, many of you will be very familiar with it, the 2-sample t-test, to say, okay, we're 94% confident that that increase is not zero. There is a real increase. Now, I'm sure many of you know that the convention is we want 95% confidence and or better if we're ever going to be persuaded that we have a change. But what's 1% confidence between friends? There's some evidence that that there is a real improvement. Whether it's 0.5%, of course, is another matter. Again, I was asked to referee a debate about this between the protagonists, and what I did in this case was to actually create 2 models, very, very simple, copper recovery versus feed grade. And most of you will know that in most concentrators, particularly flotation plants, recovery generally is positively correlated to feed grade. So you can create a model by just using Excel's trendline facility and drawing a least squares line through the point. So what you see there are two lines, one for the old reagent, which is the brown points, and one for the new reagent, which is the blue points. And just visually, you can say, well, those two lines, they cross over in the middle. I mean, they're really, it's a bit of a long shot to say they're any different. And what we've done, of course, with this very simple little idea is we've actually modeled out the effect of feed grade. So why did they get an almost statistically significant difference when they did a two-sample t-test? What's going on here? But if you look at the top right, you'll see that at the high feed grades, all the points were with the new reagent. At the low feed grades, it was dominated by points with the old reagent. So yes, when they were running the new reagent, they got a better result, but it was nothing to do with the reagent. It was driven entirely by feed grade. So sadly, after 5 months of blood, sweat, and tears, they got the wrong answer. And before we move on from these examples, just a little reminder about the effects of experimental error, which we'll talk more about in a minute, but which is sitting behind all of our data. All data carries error. Here's some nice data showing the negative correlation between bond work index and the iron silica ratio for a particular ore type. There's nearly 40 points there. And you can draw a line through it, and there's a little model, and you get an R squared of 0.5, which is statistically significant, by the way. But what people tend to forget is that because of the scatter of the points and the inherent experimental error, you should be really drawing uncertainty limits about that line. And these are the 95% prediction limits. So, for example, if you wanted to predict the bond work index at an iron silicon ratio of, let's say, 1, well, the model predicts 17.5 or thereabouts. But actually, the best we can say is we're 95% sure it's somewhere between 15 and 20, plus or minus, about 5. So I think it's just a plea to not assume that a data point you have or a model that you develop is that's it. That's the truth. It's your best guess at the truth, but with a zone of uncertainty around it. And by the way, it's just quite interesting to see there's two populations of points here. You see there's ones at the end. If you remove those, you basically get a very similar model because they're on the same line, but the R squared has dropped from 0.5 down to 0.1. Still statistically significant, interestingly. And you can see there to the right the non-linear nature of these confidence intervals. They're basically parabolas. Okay, so there are a few horror stories. So let me spend the rest of the talk trying to persuade you of perhaps what we could do if we chose to. And good experimental design is underpinned by two brilliant but simple ideas. And they are, firstly, repeats. Everyone hates doing repeats because it's more work, but it's the only way you can build an estimate of experimental error into your experiment, and you have to have that estimate in order to properly analyze the data. So repeats is one thing. You don't have to repeat the whole experiment. There are various ways of being able to do that without doing too many tests. The second brilliant idea is randomization. Randomization is the idea that the experimental design should be randomized in order or space or whatever it is you're doing, so as to essentially eliminate the effect of trends in time that might interfere with your results. So if you do, one reagent one day and the next reagent the second day, there may be a time-based system that is you need to deal with, and randomization helps you do that. The key question is the effect that I've observed in my experiment real, or has it arisen by chance? And all I'm looking at is the noise in the data. An alternative way to express that is, what's the risk of me making the wrong decision? And of course, chance and risk are two sides of the same coin. So the whole of this methodology is really based on rigorous ways to deal with those questions. So let's talk about errors in data using data from what I think is one of the most extraordinary mineral processing experiments I've ever seen. This is a grade recovery curve in flotation. You'll all be familiar with it. There are 3 points there reflecting the kinetic nature of flotation. And the grade recovery curve reflects the trade-off, as we know, between the recovery of the valuable material and the quality of the concentrate, the grade of the concentrate. In this case, it's platinum. So along the x-axis, we have recovery, up the y, we have the concentrate grade in grams per ton of platinum. And it basically shows that, as we all know, that if you want a higher recovery, you pay for that in a lower grade and vice versa. And as mineral engineers or metallurgists or metallurgists, our job is actually to push that whole grade recovery curve up. So we can get our cake and eat it. We can get a higher grade at a higher recovery. That's the skill that we try and apply to our professional life. Now, the problem with doing that is to decide whether any new curve is actually a different curve to the old one, because there are uncertainties in those 3 points. What I haven't told you is that they're actually averages of 50 repeats of the whole experiment. The reason why the company concerned did this is lost in the midst of time, but I'm very glad they did, because it's a unique data set. So there you are for each of those 3 points. They are averages 50 results. And what you're looking at is a dramatic demonstration of the inherent error in any experiment. This happens to be a batch flotation test. And the spread of results that always leads to. Now, by the way, these experiments were all done as repeats. They were the same ore, same grind, same reagent, same chemistry, same operator, same flotation machine. Everything, and the assay were done by the same assay lab. So everything was the same. That's as good as it gets. And my experience of looking at data like this is that all flotation of both base metals and precious metals, interestingly, will give you a similar story if you are ever crazy enough to do an experiment like this. So that brings us back to the line, which, as I showed you a moment ago, all models have uncertainties around them. You can calculate with very simple formulae the confidence intervals around those points, and that implies a confidence zone, an uncertainty zone around the line. And now if your new grade recovery curve with a new condition, let's say, was sitting there, you'd have to say that you have not got strong evidence that line is any different to the old one. And it's driven entirely by the inherent errors in the data. So how do we deal with that? By the way, I spent some time a while back actually developing a statistical method for comparing grade recovery curves. So you can do it. You can show whether 2 lines are statistically different or not. So staying with batch flotation, there are lots of sources of error in all experiments. In this particular type of experiments, you've got sampling the original ore, feed preparation, which is grinding and everything that goes with it, assaying, the operator, machine, chemicals, time. Those last 4 you can call the flotation test. And if you do the right experiment and you lay it out carefully, you can actually calculate the proportion of the total uncertainty, which you've just seen dramatically demonstrated, the proportion of the total uncertainty attributable to each of those sources. In this particular case, ignoring the first 2, we did a quick check which showed us, interestingly, that 1/3 of the error came from assaying and 2/3 came from the flotation test. And of course, that gives you an opening then to look at your methods and see how you could improve it. Where is most of the error? And therefore, where should we put most of our effort? So we have to measure experimental error. How do we do this? Well, there's lots of ways, but the classical way, of course, is something well, the standard deviation, which is given by that formula, which you're all familiar with. In passing, so it's a sum of squares divided by n minus 1, which is called a degrees of freedom. That's actually a variance, and you get the standard deviation by taking the square root of the variance. In passing, it shows you why a sample size of 1, n equals 1, is not a good idea, because that leads you to having to divide by 0, which, as we all know, we're not allowed to do. So by definition, if you only have 1 of everything rather than repeats, you can never determine the experimental error. So let's look at a couple of solutions to some of these problems that I've been talking about. If you're looking, and again, some of you will be familiar with these, if you're looking at comparing two conditions, then the trick is to set up pairs of experiments in which one of the conditions is measured in one half of the pair and the other with the other half. So you could think of that first little diagram there as perhaps two parallel lines in a concentrator. Maybe you're looking at two grinding lines, and you might be trying different control systems or different cyclone dimensions or whatever. And what you do is you set up, perhaps day 1, you would have your pair arranged like that. So the top one of the grinding lines is running with the condition on, the new condition, the other one with the condition off. And then at the end of that day, you collect the data and you get each result, and you look at the differences between the two. The next day, because of the need for randomization, you toss a coin. It's actually literally all you need to do to decide which way round those two are, and that's a switch. Next day, you toss the coin, don't switch. Next day, you toss the coin, switch again, and so on. So the process is randomized. And by the way, if you haven't got a parallel circuit, or if it's difficult to pair your experiments, then you can do it in series where you've got little blocks that are arranged similarly. And then you look, as I say, at the differences. So you have a population or a sample of differences, and there's a little simple statistical routine called a paired t-test, and all this stuff's available in Excel, which allows you to make a judgment about whether the differences you observe are statistically significant and what the uncertainties are in them and so on. So let me show you a real example. So again, we're looking at flotation plant because most of my examples were obtained in flotation. So this is a gold specific reagent they're trying. So along the x-axis, we've got time. Up the y-axis, we've got recovery. And I've simply plotted, the experiment was done like that top one there. So I simply plotted the two lines recoveries as they came up in the from day to day, and you can see straight away how much noise there is, and you're all familiar with that. This doesn't come as any surprise to any of you, I'm sure. It's really noisy. You've got trends, you've got cycles, you've got mess going on. Well, if you look at the average difference over the 50 odd days, 60 days of the experiment, it was 1.8%. Well, that's a good result. Anyone in a flotation concentrator would be happy with that. But can we really say that is a real difference? Because the actual day-to-day recoveries go from less than 60 to more than 80%. So is 1.8% real, or is it just buried in the noise? Well, the whole point of a design like that is that you can do a very simple little paired t-test available in Excel, and this is what we can learn from this simple test. Number one, we are more than 99% confident that yes, there has been an increase in recovery. The increase is indeed 1.8, but we can actually put an uncertainty around that. I happen to put a 90% uncertainty, plus or minus 1.1%. And this number here, which nobody ever calculates, and I don't know why, because it's very easy and it's really important. The words say, the language is that we're 90% confident that the increase in recovery is at least 0.9%. That's the worst case scenario. Everything else is an upside. So if the new condition costs you 0.9% of recovery, then it's a no-brainer. You should switch because you will almost certainly get an improvement. And that's a really useful number. And finally, the conclusions are independent of the time-based variation, which you can see is extreme. because we've subtracted it out with pretty much the simplest thing you can do in arithmetic, which is subtraction. Just in passing, we've all been troubled by experiments that didn't do what they were supposed to do. And I guess I plead to people, if the result is disappointing, don't do what I've seen a troubling number of times, which is remove inconvenient data until your hypothesis is satisfied. I think the right thing to, I'm sure none of you would do that, of course, the right thing to do is to keep calm and don't shoot the messenger, and then go back and have a think about what went wrong and what you learned from it and so on. Here's another interesting, it's one of my favorite data sets. This came from a very large new mining project in Western Australia. It was a nickel project. And it was one of the very first geometallurgical studies to be made. And they decided they wanted every block delineated in the mine to have a flotation recovery attached to it. And this involved a huge number of flotation tests. So they commissioned 3 commercial laboratories, and between them they had 7 operators doing this work over many, many months. So it was quite a complex flotation procedure as well. And I was involved a bit in the design, and I said to these guys, look, you're going to be very dependent on these operators because we don't know if they get the same answer. How well trained are they? They've been trained in this method. So I suggested that what we did was put aside a block of ore and then use the same ore to push through anonymously samples so that early on, each operator had actually done a few repeats of the same ore, and then we could just look straight at their repeatability. So here's the data. So by the time we'd finished that phase of the work, every operator had done 3 repeats and a couple of them had done 4. So if you look at the averages, look along that bottom row, it's remarkably good. 90, 92, 88, I'll drop that for the moment, 91, 91, 89, really good. But the obvious outlier is that one, 79. However, if we're playing the game properly, you'd have to say, well, I need to be persuaded by a proper statistical process that that result is statistically different to the rest. We eyeball it, yes, it looks different. The correct way to do that is using something called a one-way analysis of variance, again, available in Excel. Don't worry about all the stuff on the left. This is Excel's actual output. The thing we want to look at here is the p-value, and many of you will be familiar with p-value, so forgive me for laboring the point. But it's basically, a way to think about it is it's the risk of being wrong in saying that the operators get different results. If that risk is very low, then we can draw the conclusion they get different results. Another way to think about it is to convert it to a confidence by taking 1 minus that figure and multiply it by 100. So we are super confident that there is a difference. So it's quite clear that 79 is the outlier. It turned out they went and talked to the operator, and it turned out that he or she had misunderstood one step in the process of the flotation testing. And when that was corrected, then they came back onto the same level as everyone else. And of course, the way to doubly check its operator 4 is to do that little exercise again with a one-way analysis of variance and just leave out operator 4. And if you do get that, the p-value is about 0.5, no significant difference. And all of this is so easy to do. I mean, all of that would take you 2 minutes. By the way, there's some free information here that usually is in well-planned experiments. If you look at this MS, that stands for mean square. The MS of experimental error is actually the variance of the experimental error when the operator effect is removed. So that's a variance. And as you know, if you take the square root of that, you get the standard deviation, 2.8. And the properties of the normal distribution allow us to say that any one result, let's look at the one top right here, 90%, any one result has an uncertainty of about 2 times that figure, 5.6%. So that is 90% plus or minus about 5%. That's a lot. And most people who do flotation all the time are quite shocked at how uncertain each, and that's from a good experiment. This is not, they're not making mistakes. They're not poor experimenters. This is a good experiment. It's as good as you get. So how can you reduce that? Well, the answer is by doing repeats, because the standard deviation is reduced by the square root of n-1 every time you add another repeat. So there you go. So let me say a little bit more about factorial design, which are the gold standard. And that even that little paired trial I showed you is a kind of factorial. Definition is every factor is investigated at every level of every other factor. And you saw in the SciTech experiment a little bit earlier, a cube. Well, let's look at 3 factors, 3 variables at 2 levels. So a very simple experiment, just a low level and a high level. And that means there are 2 to the 3 runs, 8 runs. And you can visualize this as a cube, which has 8 corners, and you do an experiment at each corner of the cube. So here's some real data from a lab flotation test looking at the effect of grind, copper sulfate as a promoter, and 2 different ore types. I'll just skip that. So in this case, it was pentlandite, so it was probably a nickel-bearing ore, and the experiment was done as we've seen. Of course, we're doing 2 levels, so you're only getting it linear effects, but they are often enough. And they did some repeats at the center of the experiment. So this little block here, this little point here, is called the center point, and you do runs at that point. So the results look like this. So on the left-hand side, you've got the factor, the variable, ore type, grind, copper sulfate. So when you've done the analysis, the statistical analysis of the factorial, were those statistically significant? In other words, did they affect the outcome? Yes, yes, and yes. What was the magnitude of the effect? Well, in the case of ore type, there was a 13% difference in recovery for the 2 ore types, other things being equal. What about the grind? The grind overall actually reduced the recovery. So that's not what we normally see, because we normally grind to liberate, but there was something else going on in this, which I won't go into now. And did copper sulfate help? It did, 5% improvement. Then there's some other strange things down the left-hand side, AB, AC, BC, ABC. What are these? Well, these are the interaction effects, which if you don't do a factorial, you won't see, and they always, nearly always occur in complex systems, and as we all know, to our cost, mineral processing systems are complex. So an interaction occurs when the effect of one variable depends on the level of another variable. So it's quite a complex situation. So in this case, the copper sulfate, the way we interpret this is that the copper sulfate, so BC is the interaction between grind and copper sulfate. The copper sulfate has little effect at a high grind, so with a fine product, but a strong effect at a low grind. And if you don't do this kind of experiment, that just comes out as experimental error and it muddies the water. But we can actually tease that out as a real effect. And that was the only interaction, as you can see, that was statistically significant in this experiment. So it's very important to be able to do that. And of course, what comes out of it is a very simple linear model, recovery as a function of the 3 factors that are significant, plus the 1 significant interaction. So again, the analysis is simple and straightforward. And you can see some of that effect in these kinds of graphs, which I'm sure you all use. This is actually quite interesting because it does show the interaction effect very strongly. So at the low copper sulfate level, low level, the grind up this way has no effect. At high copper sulfate, the grind has a strong negative effect. So it's a lovely visual description of an interaction effect. So what are the advantages of factorials? Firstly, it gives the maximum efficiency in the estimation of the effects of the variables. So that's actually a mathematical statement, but it's very important that the results we get are mathematically optimal. It can detect interaction. I've labored that point, I think. This is a very useful feature. A statement about the effect of one factor can be made independently of all the others. So a factorial teases out all the effects as independent attributes of the system. The conclusions hold equally over a wide range of conditions, and the significance of the effects, as you've seen, can be determined with statistical rigor. So are they real, or is it just noise? Now, I said to you that what experiment we just looked at was two-level, which means we can only look at linear trends. But in many cases, obviously, you're going to expect curvature in the system. And so then we move to response surface designs, of which the one that we use most often is the CCRD, Central Composite Rotatable Design. Terrible name. People often mistake it for a counter-current decantation system. And a CCRD basically takes the original simple two-level structure, which you see on the screen now, and adds some points to it. And it's very prescriptive as to where those points should be to get the optimum efficiency in the result. But it prescribes the experiment. You do the experiment as designed, and you end up with a superb product with a response surface model that describes the complexities of all the effects. So it's a model that has linear terms, squared terms, cross product terms representing the interaction effect. So here's a little example, some nickel flotation, looking at the effects of frother dosage, collective concentration, and air flow rate. And there's the model that comes out of it. So the linear terms, the squared terms to represent curvature, and the cross product terms to represent interaction. There are the coefficients of the model. It will tell you which coefficients are statistically significant or not. So here's one collector square that's probably not significant. And we get a beautiful model that allows, it's obviously an arbitrary regression model, but the purpose is to allow you to predict the recovery in this particular case as a function of the different conditions you might be interested in. R squared of 99%. The S is the standard error of the regression. So you multiply that by 2, and that's the 95% confidence interval approximately of any one prediction of the model. So this model will predict recovery to plus or minus about 2.4%, which is very, very good. And there's the actual versus the predicted nickel recovery, and you can see it's a very nice result. And yes, by the way, that is the correct way around to plot them for reasons I won't go into now. And you can draw a 3D graphs and show people in a nice visual way how the system works. And this is another one that just illustrates that you can do these experiments in plants. The one I just showed you was in the lab. This was done in a PGM plant in South Africa some years ago on a single cell to look at the effects of air rate and level on grade and recovery. So grade, con percent solids is a proxy for grade. That's the one on the right. And solids tons per hour is a proxy for recovery. And you can see the effect. So with the con percent solids, we can see again that very strong interaction effect. So at a high level, air has little effect, but at a low level, it has a very strong negative effect. How do we estimate experimental error? Well, there are lots of ways. I've touched on it already. Repeat the whole experiment is the gold standard. So when pharmaceutical companies are doing clinical trials, they repeat the whole experiment several times. Or you can repeat some tests, the center point runs that I alluded to. If all else fails, really high order interactions are very unlikely to be real, so they're almost certainly reflecting error. So you can use that. You can borrow from history. And remember, blocking in time is always useful. This is the way of arranging the experiment. Generally doesn't require any more tests, such that any time effect, or whatever time is a proxy for, can be firstly assessed and secondly removed from the analysis of the data. It's really useful. So when I'm helping students with experimental designs, we always try and block in time if we can. So I'm nearly done. Stats is, in every sense, risk analysis. The p-values are risks, and we're managing the risk of being wrong in making a conclusion from our experiment. So this is a managed risk. This is an unmanaged risk. I guess we'd all prefer the managed risk, and I think I see statistics as putting the bars on the cage. So what are the takeaways? Good experiments get the right answer with the minimum of effort, with a known confidence and a known risk, so we're managing risk. Bad experiments get the wrong answer, no answer, which is quite common, or the right answer but take too long. Statistics allows us to manage the risk in decision making based on uncertain data. So I was once involved in some plant scale experiments on the introduction of new grinding technology with a company with several concentrators. We did a proper paired experiment. It was going to be $100 million investment or some large number like that. And we were able to tell the authorities and ultimately the board that we were very confident that this new technology would improve recovery by at least a certain number. I think it was about 2%, which would more than pay for the investment. And so, in a sense, it was a no-brainer. We had done a good experiment and we'd allowed the board to de-risk their decision-making. If you do the right experiment, the analysis follows. It takes care of itself. So I hope you've been convinced that stuff is powerful, but it really isn't hard. And if I can refer back to how I started, everybody else does it, so why don't we? It's all about how to make wise decisions in the face of uncertainty. And Mark Twain is said to have said this. There's some dispute about it, but why spoil a good story with the facts? Collecting data is much like collecting garbage. You must know in advance what you're going to do with the stuff before you collect it. And I'm going to finish with pictures some of you may have seen before. One of my favorite pictures. This is literally a plant trial. It's a trial of plants. It's a trial of trees to be grown for telegraph poles. And it is one of the very first experiments ever to be done under the direction or suggestion of R.A. Fisher when he was working for the Rothamsted agricultural station that we touched on right at the beginning, looking at the variety of conifer, and the distance up the hill. This is in North Wales, and I believe some of those trees were only finally cut down in the 1980s. So you can see the randomization here. Do you see these 5 different species of tree or varieties? There's the next level, and this light colored one is here. So they've randomized the order. So what they get at the end of it is which is the best variety and what is the effect of the altitude of the hill and the different degrees of soil fertility that is represented by that. So it was a brilliant experiment. One small problem, it took many years, so I don't know quite how you hold on to your professional career while you're waiting for that experiment to finish up. So that's it, folks. I hope I've made my point, and I'd be very happy to answer questions if you have any.
Thank you very much, Professor, for the interesting presentation. So we have time for questions. I think someone has raised a hand. Alexei.
Hi, Prof. Thank you so much for a very interesting presentation. My question relates to the design of experiments, specifically for the mineral processing plants. There is a big hopes of literature on optimal designs, like D-optimal, I-optimal, etc. Do they make sense for what we're trying to achieve, or is it overkill?
I don't know enough about them to answer the question really, but I have read something about them. My feeling is the trouble is I'm getting old, so I think everything that was done 20 years ago is as good as it can get. And clearly, we do make progress, even though that book I mentioned is 100 years old. I think my gut feeling is that it's overkill because these designs are very simple. They're not complicated. They're very powerful. And most people who work in concentrators are not, if I can put it this way, academically the top rank, which are the people I'm talking to today. They need, I mean, they're smart as tacks and they're very good at what they do, but they need a way of doing experiments that they can understand, that is not complicated, that is not demanding, because they've got other things to do. And this is why I often say that I think we have to take it into our DNA. So it's not controversial. The only way to do that is to make them easy to do, so you do them all the time. And again, I come back to all the other people that the other professions and industries that I mentioned at the beginning. They are using these sorts of designs. I'm not sure the extent to which they've moved to other forms of design, but my wife, actually, I shouldn't quote her, but she's actually a pharmacist. She's spent a lot of time working on clinical trials. So that's where my knowledge of that comes from. And they are essentially classical designs of the kinds I've been talking about. So I'm not sure if that answers the question.
Right, so thank you. And the next one from David.
Yes, Tim, thanks for your presentation. We're in the Centre for Minerals Research are sold on statistical design. We've got 3 of your textbooks, and we use Minitab, which I think was your recommended product that we use in MP Tech. So I've no doubt of the importance of statistical designs and their interpretation when it comes to industrial stuff. My question is a little bit broader. So in the past, we used to have metallurgists who knew nothing. And then eventually we got metallurgists who had certain tools to understand what's happening on their plants. And now we've moved into an era where, well, I checked out on a survey, 70% of students in the world use ChatGPT and AI routines, okay, in attempting to analyze data. And then you've got data analytics running through. So somewhere you've got this match between physics, chemistry, metallurgy, statistics, and then the interpretation of them by various platforms. And my colleague Martin Harris, whom you know well, he insists that you have to understand the underlying physics or chemistry before you can make any sense of a situation. I'm not sure I agree with him, but what are your views?
Yeah, it's a great question, Dave. I really enjoy the question. And I've thought long and hard about AI and what impact it's going to have on all this kind of stuff. So again, I'm no expert, but my understanding is that AI, you can think of it as an incredibly powerful device for essentially sweeping the internet, taking all the stuff off it, and then presenting it and using it in a very useful way to convey things to human beings. So, for example, somebody said to me a year or two ago, look, we don't need all this stuff anymore because we just get all the data off the plant, stick it through an AI system, and out will come the answer. Well, out will come an answer, but because it's based only on the data that the concentrator produces, it will never cover all of the operating conditions that you would need to get a full understanding of what the process really does. It's called happenstance data. I'm sure you know, it's a technical term. And happenstance data, especially in a concentrator where you are running it to very tight constraints, like a minimum concentrate grade for the smelter contract and so on. So it's not an experiment that has any rigor to it. So at the moment, my feeling still is that this kind of stuff and stuff like it it will always beat happenstance data that just comes out of the process, willy-nilly. But AI is, as we all know, developing incredibly rapidly, and it's incredibly powerful. And I think it would be a brave man or woman to make predictions about what might happen in our game. By the way, Minitab, you mentioned good old Minitab. The only reason I recommended it, it was recommended to me by a real statistician who I worked with very closely when I was working at De Beers for many years. She was great. She taught me a lot. She used Minitab, so do I. I don't know if that answers the question, Dave.
Yes, thanks, Tim.
Thank you. Professor Marcelo.
Tim, fantastic presentation by the master, and I'm also an enthusiast of statistics and experimental design. My question is a very short one. In the beginning of your presentation, you gave some examples of some poor or lack of use of statistics in important plant testing. And one area that I thought is still kind of a blind spot in people is the fact that sometimes, let's say that you're comparing reagents, like you talked about in the beginning, the copper ore. And let's say the new reagent is not meant to be better, but it's meant to be cheaper. So it's really cheaper. And actually what you want to prove is that there's no loss in recovery by using this reagent. I think very few people, I'd like to hear your comment on, don't really realize on the type 2 error in this case, because sometimes you kind of say that, yes, there's no difference in these guys, whereas actually the new reagent or cheaper could be much worse. But if the experiment was not properly done, it could be a disaster. So I'd just like to hear from you, because many more people are using this, but fewer people really grasp this type 2 error, which is a little bit tricky. Please.
Yeah. Another great question, Marcelo, as I would expect from you. Absolutely agree. And of course, there is, if you do the correct experiment, a paired type of experiment, there is a slightly different way to analyze it using a t-test, which looks exactly at this point and gives you a decision as to whether whether you can, within defined limits, conclude that the two conditions are the same. Because you know well that the whole principle of the null hypothesis is you start with saying that they're the same and then reject that hypothesis. This does it the other way around. It's called the 2-1 sample t-test. You do two t-tests the other way around, and you end up with a conclusion as to exactly what you're saying. If you can demonstrate that they're not different, you can't really say they're the same. They're not different by a defined amount, but one is much cheaper than the other, then you have the basis on which to go. And in fact, again, pharmaceutical companies do very similar experiments. They want to bring in if they are a generic company, they want to prove that there's no difference in performance between theirs and the market leader. So yeah, great question. All down to type 2 errors, and but again, there are simple solutions.
Thanks, Tim.
All right, so we have one last question from Michelle.
G'day, Tim. Let's get rid of that hand. So, great presentation. I think you've done this before. So, I was wondering if you could help me name something. I've done an analysis for a job, I don't know, it was a while ago now, but I've been wondering what the hell to call it. I used an ANOVA statistics experiment as the foundation, and I used a stochastic simulation on top of that. So what we had was there was a deposit that had was controlled by mineralogy, and the two control vectors was percent carbonate, and the other one was percent magnetite. So I had an ANOVA panel 5x4, and on each of the panels I had a batch grind, I had a flotation test, and I had an SMC test, which I then put through a Monte Carlo stochastic simulation in a formula, using those as the basic foundations to work out throughput. So it's a geomet job. What the hell do I call that?
Oh,
Like, is there a name for
I
that
don't
sort of
know,
analysis?
but I'm not in the naming game, Simon. But if I could just sort of fire this one back at you and say, you know, mixtures of this kind of thing, I think are really good and powerful. For example, we have done with students here, and I'm sure many of you may have done the same thing. We've used experimental design to actually run a very complex simulator to produce a much simpler usable model. So if you've got a very powerful but complicated simulator, one approach to making that available in a simple way is to actually get it through an experimental design to predict outputs as a function of inputs, and then model that in a very simple way. And so you've got a sort of combination of experimental design with a simulator. So nobody actually does any experiments. We take an existing complex simulator and simplify it, if you like. It's not quite what you've done, but certainly we've done. I mean, we all know. I'm sure you all know better than I do. Monte Carlo type methods are really powerful, and I've used them quite a lot in this stuff. And so what you've done sounds good to me, but you're the sort of guy who can name things, Simon, not me.
Yeah, well, I was trying to describe it simply. Yeah.
Well, we might have a conversation offline.
Incidentally, my students in Belgium, I used to after doing your course a few times and reading your work, I used to bash my students with experimental design, but I used the words, what is your plan? And what does it mean if you're successful? How do you tell the difference? So, anyway, there you go.
And what's plan B when you fail? That's the other thing. Look, before we go, I should apologize to many of you who are persuaded of this stuff. Marcelo and Dave and the rest of you. I know that, and I know I was preaching to the converted to some extent. Aubrey asked me to give a talk. I said, no, I've given up giving talks. I've got nothing useful to say anymore. I said it all. He said, oh, no, do that one, you know, that you've done before. So you can blame him.
Well, thank you again, Professor, and let's thank you for the very interesting presentation once again, and thank you all for attending the seminar today.
Tim, you don't have to apologize. You don't have to apologize for anything, Tim. It was a good talk.
Thank you, Dave.
Thank you very much.
Okay, well, goodbye. Thank you. It's been lovely seeing you all.
Thank you.
See you.
Yes, everyone.
Thanks a lot,
Bye.
Tim. Bye.
Thank you.